guys, I'm an educator Sulagna Ghatak. Today I gonna brief you about the topic DNA recombination. So let's start. So DNA recombination is the process that involves the rearrangement of genetic material which takes place by means of breaking of genetic material and then rejoining of genetic material. Here is the DNA molecule. So in eukaryotes, crossing over or which is the physical exchange of genetic materials occur in prophase 1 of meiosis, and which is also governed by mechanism of recombination. And in prokaryotes, recombination occur in conjugation, transformation, transduction. During the integration of transfer of genetic material into bacterial chromosome reciprocally. So there are mainly two types of DNA recombination processes: homologous recombination and site-specific recombination. So let's see what is homologous recombination. Homologous recombination is a type of genetic recombination that occurs during meiosis or during the formation of germ cells, that means the egg and sperm cells, right? So the paired chromosomes from male and female parent align contains similar DNA sequences from the paired chromosomes and which cross over each other, and crossing over results in a shuffling of genetic material and is an important cause of genetic variation which can be seen in the offspring. So there are few models which explains the mechanism of homologous recombination, and among them two are most important: holiday model and double strand break model. So let's see what is holiday model. So holiday model of recombination is also called heteroduplex model, and it is the widely accepted model for DNA crossover, and it was first coined by Robin Holliday in the year 1964. So let's see the process of holiday model. First of all, two homologous DNA molecules line up. Then cut into one single strand in both duplex occur. Then clipped strand cross and join homologous strand to form holiday junction. So this is the holiday junction. And lastly, heteroduplex region is formed by the branch migration. This is the heteroduplex region or hybrid duplex. Okay. So there are few important proteins and enzymes for a holiday model. In E. coli, recombination is initiated by the enzyme REC-BCD, which is the main enzyme, and it consisting of three subunits: REC-B, REC-C and REC-D. REC-BCD enzyme has both helicase and nuclease activities, and also REC-A enzyme has a vital role in case of recombination. It can wrap around single-stranded DNA and direct it to form holiday junction. Branch migration is catalyzed by RUV-A and RUV-B proteins, and at last holiday structure is released by RUV-C protein to form a duplex DNA. Okay? Now let's see the enzymatic actions in holiday junction. So first of all, REC-BCD enzyme uses its helicase activity to unwind DNA duplex. Then, after unwinding, REC-C recognizes the chi site into the DNA. And why it is called chi site? Because Greek letter chi is look alike a crossover point. Then, REC-C signals REC-D to stop. And when stopped by the help of ATP, REC-D signals REC-B to cut by its nuclease activity and continue unwinding of DNA. Then REC-A become loaded and REC-BCD dissociated. After that, two processes become occurred: reciprocal break joining and non-reciprocal break joining. And this non-reciprocal break joining is not actual type of recombination. Okay. Now, the second type of DNA recombination process is site-specific recombination. Site-specific recombination is also known as conservative site-specific recombination. It is a type of genetic recombination in which DNA strand exchange takes place between segments possessing at least a certain degree of sequence homology or common sequence. Enzymes known as site-specific recombinase or SSRs perform rearrangement of DNA segments by recognizing and binding to short specific DNA sequences or sites at which they cleave the DNA backbone, exchange the two DNA helix involved, and rejoin the DNA strands. Site-specific recombinase or SSR are highly specific, fast and efficient even when faced with complex eukaryotic genomes. They are employed naturally in a variety of cellular processes, including bacterial genome replication, differentiation, pathogenesis, and movement of mobile or transportable genetic elements. Now let's see an experimental proof of site-specific recombination, and in this case, integration of bacteriophage lambda DNA into E. coli genome involves the site-specific recombination. Bacterial genome BOB' sequence contain the 23 base pair, which is called ATTB, and also bacteriophage genome or lambda genome POP' sequence called ATTP contains 250 base pair. Each one genome comprising an identical or same central sequence O, which is 15 base paired. And recombination event is catalyzed by lambda integrase, which is a tyrosine recombinase and which can break phosphodiester bond and covalently attach to 3' phosphoryl group, which we have seen in case of type 1b topoisomerase in the process of replication, right? So here IHF or integration host factor bends more or less 20 base pair sequence in ATTP and bring together the lambda integrase binding site on DNA arms. And lastly, hybrid strand become formed consisting of BOP', which is called ATTL, and POB', which is called ATTR. So, hope your concept about this topic have been clear after this session. There has provided a link of e-quiz into the description box, so you can score healthy marks and test yourself by those questions. And if you have any doubt or facing any problem during solving those questions, then after scoring you can ask me into the comment section, and also you can reach to us through our mail ID, which is into the description box. I'm so much happy to say that in previous lessons I've got your lots of responses and doubts through mail, so don't miss the chance to get a healthy score. Lastly, thanks for watching and don't forget to subscribe our channel, then you can get notifications about my next videos which is coming on every Sunday. So stay safe, stay tuned. Bye.